teach you fun facts or teach you something new that maybe you didn't know. Yeah, so we have another one today. I was definitely at a loss for what to teach next for a while, but now that I am in grad school for genetics, I'm learning genetics all the time, and so I constantly have new ideas of what to teach. So today we are going to be talking specifically about epigenetics. Epigenetics. And if you don't know what that is, don't worry, we are going to talk all about it, but just a high-level overview is that epigenetics, the prefix epi, means like on top of or like accessory to. And so you have your genetics, you have your DNA sequences that are in every cell of your body, and you can't change that. That's how it is. That's your DNA that you got from your parents. But epigenetics refers to essentially modifications that can be made to your DNA that impact how it is expressed or where it is expressed or when it is expressed. So yeah, it impacts which genes in your body are expressed where, and that is a completely normal thing. It has to happen in everybody, but it can also lead to like diseases, or it shows how like lifestyle factors can actually impact your DNA or impact your children's DNA. We will talk about that. So yeah, if it doesn't make sense, don't worry, we're gonna get into it and it will make sense. So first I want to get started by talking a little bit about DNA transcription, which I feel like is kind of an important topic to set the scene for how epigenetics works. So I talked about it in my last video a little bit, but I'm going to go over it again, potentially with some focus on different things that are important to this video. So up here we have our DNA sequence, our double helix of DNA. That's what DNA looks like. But DNA does not make you who you are. It writes the instructions for making you who you are, but you have to actually transcribe your DNA, express your DNA to make you who you are. So I kind of like to think of it like all of the DNA in your body is like this big bookshelf of cookbooks. You have all these cookbooks in your bookshelf, but it doesn't mean that you have every food that is listed in those cookbooks in your house. You don't have every dessert, every meal that's in those cookbooks in your house. You just have the instructions to make them if you want to. So that's kind of like this. Every cell in your body has the whole bookshelf of cookbooks, but certain cells of your body express certain genes or make certain desserts, if you will. So like your bones express genes that are related to bone growth, making bone. So how that happens, how a gene is expressed or transcribed are like the words I will be saying a lot in this video, is that the DNA has to be opened up. So it's not a double helix anymore, it's opened up so that you can see each strand of that DNA. And what we're showing here is the 4 nucleotides. So you have thymine, T, guanine, G, cytosine, C, and adenine, A. And you can see all of those there, and I guess I've drawn thymine as green, adenine as pink or purple, cytosine as yellow, and guanine as blue. And so here we have a particular gene. Any gene in your body, you have like 20 to 30,000 of them. Yeah, and so where that gene is, next to the gene, we've got what we call promoter sequences. And the promoter of a gene essentially shows that promoter sequence isn't actually part of the gene, but it helps to recruit all of the proteins that are necessary to transcribe the gene. So all of these things called transcription factors and other like proteins, RNA polymerases, they all come and they recognize these promoter sequences, and that tells them, hey, there's a gene here. We need to express this gene. So these are actually the particular sequences. This one is 35 nucleotides upstream, or like before where the gene starts, which is here. And then this one is 10 nucleotides ahead of where the gene starts. A nucleotide is like one of these letters, essentially. So yeah, this one's called the Tata box because it's T-A-T-A. So yeah, try being a middle schooler and learning about the Tata box in a room full of middle school boys. Fun times. But essentially, those are the sequences that are recognized, and then we get this mRNA that is transcribed. So we've got DNA, and we transcribe an mRNA strand that is complementary to the DNA, meaning T always goes with A, G always goes with C. So like anytime you see a T, we've got an A on the complementary strand. mRNA does have uracil instead of thymine. Kind of confusing, but yeah, uracil is what binds with adenine or A instead of T in mRNA. So you can see that we have a new color. We have red. And anytime we have an A or a purple in the DNA, we get red or a uracil in the mRNA strand. But not super important for this video. The main thing that I'm trying to tell you is that DNA has to be opened up, has to be stretched out, and all these proteins have to be able to come in and actually transcribe that DNA for it to be expressed in your body, for you to make that cupcake that's in the cookbook. All of this has to happen. But yeah, not super important to this video, but the mRNA that is made there is then down here. It's basically like a copy of the recipe. It's like getting the recipe out of your cookbook, and each 3 nucleotides of that mRNA strand codes for a different amino acid, which are the building blocks of proteins. So this is taken and it's made into a protein, which now we're talking like the actual food, the recipe. So we get like a protein, and that does whatever it does. Maybe this protein is a liver enzyme. It's expressed in your liver and it helps you digest alcohol or something. I don't know. But yeah, different proteins expressed in your body from the genes. DNA has to be opened up and expressed to make genes. So that's like pre-epigenetics. We're setting the stage, and now we're going to talk about how DNA is organized in your body and kind of where epigenetics comes into play. So over here we've got our double helix that we already talked about. You can totally ignore these red doohickeys for now. We're going to talk about them more on the next piece of paper. I almost said slide, like I'm a teacher giving a presentation. Next demonstration piece of paper, but you can ignore them for now. So this is our DNA strand, and how it is actually organized in your body, in your cells, is that the DNA is coiled up and it's wrapped around histones. So the blue doohickeys are called histones, which are specific proteins, and this is the structure of a histone down here. So that shows what a histone actually is. It's the proteins H2A, H2B, H3, and H4, and each histone has 2 of each of this would be called euchromatin, or euchromatin, euchromatin. And that's more like loosely organized. And then all of that is like packaged into a chromosome. You've probably heard of chromosomes before or seen them, so yeah, but that's what the DNA actually looks like when it's packaged into a chromosome. And I just want you to think for a second about what we talked about before with DNA being opened up and transcribed by all these proteins. Which of these organizations, this or this, do you think would be more conducive to expressing DNA? Like, which one is going to have genes that are going to be transcribed? If you said this one, you're right. Why? Because it's all open. It's loose, it's open, and so those proteins can get in there and they can transcribe the DNA. And so for places in your body where a gene is expressed, it's going to look more like this because those proteins can get in and they can express the DNA. Whereas here, this would be more silenced. Those genes aren't really being transcribed because this is super compact. It's all compacted and the proteins can't get in. So we're going to talk in the next part about how that happens. How that actually happens and how it controls the transcription of your genes. Okay, so this is definitely the piece of paper I'm least proud of. I think it's kind of ugly, but ignore it. So the two main epigenetic marks that we are going to talk about are DNA methylation and histone acetylation. There are more. There are like ubiquitination and phosphorylation and all that crap, but we're not going to talk about them. I haven't learned that much about them, and they're not talked about as much. But over here, we're going to look at this first, and then we'll look over here. So over here, we've got DNA methylation. And if you remember me talking in the first part about promoter sequences, those sequences that are upstream of the actual gene, those can be methylated. And so methyl groups are, it's like a carbon with 3 hydrogens, I think, a methyl group, and it can be added on to cytosine nucleotides in DNA. So the C's, the ones that were yellow in the first piece of paper. So those methyl groups can be added onto cytosine. Oftentimes it's in the promoter, but it can be in the gene itself. And what that does is, now imagine those proteins coming in to transcribe the DNA. Well, they can't get to it. They can't get to it because it has methyl groups on it. So here, the proteins can't get there, the transcription factors can't transcribe, and so you're going to get a gene that is not expressed. The DNA is not going to be transcribed, and this gene is inactive. Whereas here, if there's no methylation on the promoter, it's nice and wide open, and the transcription factors can come in and express the gene. And so this one is active. I'll go ahead and talk about this, and then I'll talk about like what that means or stuff related to that. So yeah, this would kind of be associated with chromatin that looks like that, that's really compact and tightly wound, and this would be associated with less compact chromatin. So, okay, so now let's talk about histone acetylation, and then we'll go back. So this is the heterochromatin that's really tightly packed and is not being transcribed. And the reason it's so tightly packed is because DNA is negatively charged. It has a negative charge on it, and histones are positively charged. And so what happens when you have a negative charge and a positive charge? Opposites attract. Opposites attract, and so you get DNA that is really tightly wound. It wants to be around those histones. DNA sequence, acetyl groups are added to the histone tails, the tails of those histones. And what that does is it neutralizes the positive charge on the histones, and that makes the DNA kind of release, because it's not as attracted to the histones anymore, and it kind of releases, and it makes it more loosely packed, and where those proteins can come in and express the DNA. So that would be active. So it's kind of opposite, where like if you have this, you're not going to be transcribed. If you have this, you are going to be transcribed, or you are going to be active. So the main thing to know about this is that these marks and every epigenetic mark is reversible. It can be added or taken away throughout your life, and so that has a lot of impacts. Obviously, not all of it is like going to be reversed, but epigenetic marks as a thing are reversible. Sorry for my stomach, oh my gosh. So yeah, they are reversible, and so epigenetics is very important in your body. It's very necessary. It needs to happen. The main thing I'll say is like every cell in your body has all of your DNA, every gene. But we don't want to make liver enzymes in our bones, and we don't want to make bone in our liver. That'd be really bad. And so in certain cells, like in your liver, those genes that make your bone or those genes that make your hair are going to be inactive. They're going to be methylated, and hopefully that'll stay that way throughout your life. Whereas like the genes for liver enzymes are going to be active in your liver. But it can also change over your life. So your lifestyle factors can impact your DNA expression. I actually did a project on how sugar intake, like eating too much sugar, can lead to the hypermethylation of a gene. So it leads it to be not expressed, to be inactive, and that can lead to Alzheimer's disease. So you can get Alzheimer's from eating too much sugar, and the way that that happens is through epigenetics. There are other mechanisms by which it happens, but like epigenetics plays a role in that. So epigenetics can also change throughout your lifetime. Just regular aging is associated with more or less methylation of different genes. So that's something that epigenetics impacts. I'm trying to think of the other things that I was going to say. One thing is it can also lead to diseases if you inherit a gene that is improperly methylated, or a gene becomes improperly methylated in your body. And one example of that is tumor suppressors. So you have all of these genes in your body that are tumor suppressors. They prevent you from getting cancer by making your DNA replication go properly, or like checking to make sure it went properly. And if those tumor suppressors are methylated when they shouldn't be, it means they're going to be inactive when they shouldn't be, which means they're not going to suppress tumors. They're not going to be able to prevent you from getting cancer. And so you might get cancer because that gene is hypermethylated. So that's one way that it can cause disease. Something really cool that I want to talk about, I mean, maybe it's like frightening, I don't know, but it is how the lifestyle factors of your mom and of your maternal grandmother can actually impact your epigenetics, and therefore your DNA expression, your genes. So the way that that happens is she is pregnant can have an impact on your epigenetics. One thing that we know is like, well, if your mother drinks alcohol while she's pregnant with you, that's obviously not good. Like, it's going to lead to birth defects or things like that. But her lifestyle factors can also impact the epigenetic marks that are in both her and in you, and it can have an impact on your life forever, like have an impact on your life. And so one example of that is that if the mother is malnourished during pregnancy, if she doesn't eat enough, if she is malnourished, then that can lead her baby to have all sorts of metabolic issues. The baby can be struggle with obesity, struggle with diabetes, all sorts of metabolic conditions because of the mother's lifestyle factors, because of the mother's malnourishment. And that was actually discovered, I don't know, it was some sort of war. I don't remember what war, if it was like one of the world wars or what, but there was a particular country that had like a food embargo on it. So there was not food coming into that country, and everybody was under like a very severe famine for a year or something. And they did a study on the babies that were born to mothers that were pregnant during that famine, which meant they were eating like hardly anything while they were pregnant. And that's what they found. They found that those babies had all sorts of like metabolic conditions and were more prone to obesity and things like that because of their epigenetics and how their mother impacted them. And so going a step further, and don't worry, we will get to some cool applications of epigenetics in a second, but going a step further, every female was born with all of the eggs she was ever going to have. If you're a female, you were born with all of the eggs you're ever going to have, and those were all developed by like month 6 of you being a fetus. Several months before you were born, you had all the eggs in your ovaries that you were ever going to have, which means all of the eggs you're ever going to have were inside your mother. All your eggs were inside your mother. So if you have a child one day, the egg that created them was once inside your mother, which means the egg that created you was once inside your maternal grandmother, so your mom's mom. The egg that made you 50% of you was once inside of your maternal grandmother, which means that the grandmother's lifestyle factors, anything that affects like her epigenetics and the baby's epigenetics can impact your epigenetics. So yeah, pretty crazy, basically showing how like lifestyle factors can have a huge impact for a long time. They can affect you for generations, and yeah, affect you for generations. So pretty crazy. You were once inside of your maternal grandmother. Let that sit in. Yeah, I do have like a real example of that my aunt was born in like the 60s, I guess like 1963, and my mom said that at that time it was like a really big thing to try to gain as little weight as possible when you were pregnant. I don't know why that was a thing, but it was like, don't eat anything during pregnancy. You don't want to get fat. Like, you want to be as light as possible. And my grandmother did that with my aunt. She gained like 5 pounds her entire pregnancy. And my aunt, unfortunately, really struggles with like her weight and thing is, if you're pregnant, eat a bunch of food. That probably has an effect too, that I don't know. But yeah, so next, now that we've gone over epigenetics in general, we're going to talk about some really cool applications of epigenetics, or like how it plays a role in your body or plays a role with some diseases. Okay, so the first application of epigenetics that we're going to talk about is something I never really understood before. Like, I had learned it a lot, but it just didn't make sense. But I feel like this most recent time that I learned it in grad school, it made more sense to me. And that is imprinting. It is called imprinting. It'll be written on the next paper, but so basically, normally, like what we normally learn is that you get 2 copies of every gene, every chromosome from your mom and from your dad, 1 from each, and they are the same. Obviously, your mom and your dad don't have the same genes, but like you're inheriting a gene from your mom and the same gene from your dad, and they have the same methylation patterns. Either they're both expressed or they're both not expressed. They have the same methylation. They both work equally. Well, that is not the case in imprinting. So imprinting basically describes the phenomenon where specific genes or chromosomes are methylated in a parent of origin dependent manner. So what that means is a specific gene on a specific chromosome or a specific region of a chromosome, you always get it methylated from your mom and unmethylated from your dad, or opposite, vice versa, depending on what region it is. But you always inherit like a methylated copy from one parent that's not active and an unmethylated from the other parent that is active. And it's really important that that is the way it is, like that happens properly. Very confusing, but I will show you like how that happens, or how that has an effect in the next piece of paper. But basically what this is showing is that mom and dad, they have the same genetics. Like they each have a pink chromosome and a blue chromosome. They're the same. They have the same genetics. Obviously, again, they're not the same. They have different parents and they're different people, but like they get the same genetic material. So this is the chromosome that the mom inherited from her mom, and this is the chromosome that she inherited from her dad, and same down here, like his mom and his dad. And it doesn't matter like for this which one's expressed or whatever, because that just depends on the gene. But in this scenario, we are saying that from the mother, the chromosome you get from your mother is active. It's not methylated. So like we talked about before, it doesn't have those methyl groups. It is active. It's expressed. So both of them have that. They both have one from their mom that is active, and then one from their dad that is not active. So the dad's is methylated, the chromosome he got from his dad and she got from her dad, and the chromosome that dad is going to pass on to his baby is methylated. It's not active. It's not expressed. And so then the baby gets the same thing as the parents. They get one from the mom that is active and one from the dad that is inactive. And something that didn't make sense to me about this at first, I don't know if it's what you're thinking, like, I don't know if, yeah, maybe it depends on how much genetics knowledge you have, but what didn't make sense to me was me thinking, okay, so the chromosomes that my mom gives me are all from her necessarily this chromosome. What happens is that when eggs and sperm are being formed, so like mom has a chromosome from her mom and a chromosome from her dad, and they have the correct methylation patterns, but when she is creating eggs, those methylation patterns are removed. Because I said they are reversible, they are removed, and all of her chromosomes that she's making in the eggs are methylated in the pattern that the mom is supposed to be passing on. So all of the chromosomes she has would become active, like all the methyl groups would go away, and so then either chromosome that she gives the baby would have the maternal methylation pattern. It would be active and unmethylated. I don't know if that makes sense, but essentially, like, this chromosome that she's giving to baby could be that one. It's just that in the germline, it loses its methylation so that it has the proper maternal methylation pattern. I hope that makes sense. And the same thing happens with the dad. So essentially, yeah, you could be getting like the chromosome from your maternal grandfather, but it's just that it was like the methylation was changed when she made the egg that made you. So yeah, but that's the basis of imprinting, is basically just that the methylation pattern from each parent can be different for specific genes, not for every gene, but for very specific ones, and that can have different impacts, and so we are going to talk about that next. Okay, so specifically, we are going to be talking about Prader-Willi syndrome and Angelman syndrome. Maybe you've heard of these, maybe you know someone that has one of these, but what's really cool, kind of like the tagline about it, obviously they're not cool, it's not cool to have a genetic syndrome, but, or it, whatever, I can't say anything right. What's cool is that these people with Angelman syndrome and Prader-Willi syndrome, they have the exact same genetic makeup, but they are completely different syndromes. They lead to completely different effects. So let's talk about that. Let's go through it. Alright. So, the chromosome that we are talking about when we talk about these two syndromes is chromosome 15. And, it's just a part of chromosome 15, so it's not the full chromosome. And what's meant to be shown by these dots is that that is the centromere. So, if we go back to this one, that black dot in the chromosome is the centromere. And so this is just showing one arm of the chromosome, right? Like, it's not showing the other arm. But yeah, so we are looking at a specific region of chromosome 15 here, and these are all genes. I know it looks similar to like how I drew the methylation marks, but these are genes that we're talking about, because each chromosome has a bunch of different genes on it, so these are all genes. And you can see that in the mom and in the dad, we have the same genes. Ignore the coloring, ignore the colors of the genes, but we have the same genes. We have 5 little doohickeys with this pattern, and then we have 2 with this pattern. So the same genes that you're inheriting from your mom and from your dad. However, in the mom, only these are expressed. The ones that are colored in pink are expressed, and these that are not colored in are not expressed. So we could say that they are methylated or deacetylated or whatever, using our language that we've learned, but they are not being expressed from the mom. So this is like expressed and these are. So if you look at these chromosomes combined, like if you get one maternal and one paternal, as we all did, we all have a mom and a dad, as we all did, you have every gene expressed once. Because these are expressed here, these are expressed here. So every gene on this chromosome is being expressed in you. It's just that these come from your dad and these come from your mom. Okay? Does that make sense? And I just wrote here, this little region is called AS for Angelman syndrome, and then this one is called PWS for Prader-Willi syndrome. Yeah, so we'll talk about why. But, okay. So this is normal. You're totally normal if you get these genes from your dad and these from your mom. Now we're going to talk about Prader-Willi syndrome. Prader-Willi syndrome, I guess I can talk about what it is later. I don't know. Yeah, okay. So here, in Prader-Willi, this is the chromosome that the child is getting from their mom, paternal, paternal, mom and dad. This is the one she's getting, or it's not a girl, it doesn't matter, getting from mom, and it's normal. It looks just like that. It looks just like what you're supposed to be getting from mom. So these genes are expressed. We're all good there. But from the dad has a deletion of this region of the chromosome. Part of his chromosome is deleted that he passes on to the child. This is not a chromosome that's deleted in him. I guess it could be if you are someone with Prader-Willi that is having a child, but typically it's not. The parents are normal. But somewhere along the line that got deleted. And so what happens is that this region is totally fine. The Angelman syndrome region's totally fine because it's being expressed from the mom just as it should. And it's not supposed to be expressed from the dad, so it doesn't matter that we're missing it. But what we're missing is this part. What we're missing is this region that's we're supposed to be getting from our dad, and we're not, because it's deleted. And so there, you're not expressing the Prader-Willi syndrome region, and you get Prader-Willi syndrome from that, because you're not expressing what you need to from your dad. Here, this is another way that Prader-Willi syndrome can happen, and that is called uniparental disomy. I won't go too deep into what that is. Well, it's basically when you get two of the same chromosome from one parent rather than one from each. That you're probably like, what? Well, how that happens is that for each of your chromosomes, it's possible for meiosis to not happen properly. So basically, like, you get the non-disjunction of chromosomes where two of them go into one egg and none of them go into the other egg. And so then if the one with two combines with a sperm, you now have three of that chromosome, which is bad. If we're talking about the 21st chromosome, then that baby could live with Down syndrome. That could be okay. If we're talking about the 15th chromosome, that's bad. You can't live with three of your 15th chromosome, so that's not going to happen. But sometimes the body is pretty smart and it will get rid of one of those three chromosomes. And so imagine if the non-disjunction happened in the mom. So she created an egg with two of the 15th chromosome, and it combined with a sperm that had one 15th chromosome. So it makes an egg it makes a zygote that has two of the mother's 15th chromosome and one of the dad's. Well, sometimes the cell will say, wait, this isn't good. We need to get rid of one of these because we're not supposed to have three. And so it'll get rid of one. But you're okay on that front. It's okay because you have it, but neither chromosome is going to express this region. So we have all of our genetic material. Here we're missing some, here we have it all. It's just that we're still missing this region because it's not being expressed, and so you get the same effect as you get there with this one. And again, you get Prader-Willi syndrome. The other thing that can happen is that there can just be a methylation error. So like you could still get the chromosome from your mom and a chromosome from your dad, but like in, let's say, the dad, this region is abnormally methylated. Like for whatever reason it got methylated, and so we're getting a chromosome from each parent, we're getting the right genetic material, but we're not getting this expressed because it's methylated in both parents. So that's the third way it can happen. Those are the three ways. And so that's how you get Prader-Willi syndrome. Over here, Angelman syndrome, it's exactly opposite. Exactly opposite, where if you have a deletion in the mother, you're expressing the Prader-Willi syndrome region perfectly. Dad's not expressing Angelman region, but deleted in the mom, now we don't express this, no pink, and so we get Angelman syndrome. And same down here with uniparental disomy. If you get two chromosomes from the father, then you can also get Angelman syndrome. The one thing I will say, uniparental disomy is a lot more common for Prader-Willi for the mother, because non-disjunction happens a lot more frequently in females in the mother. Like typically if a child has Down syndrome, it was the mother's, like the child has two of the mother's 21st chromosome, not the father's. And so if non-disjunction isn't happening in the dad, you can't get a uniparental disomy with the dad. So it's less likely for that to happen. But it could still be a methylation error, just in the mom. So yeah. But just to talk about what those are, again, exact same genetic material, especially like for these two and these two, same genes. It's just based on epigenetics, based on where they're expressed, how they're expressed. We get two different syndromes, and Prader-Willi syndrome is typically people have like mild cognitive impairment or intellectual disability. They'll also have low muscle tone, and one of the main symptoms that's really terrible is that they have the inability to feel full. They will eat and eat and eat if they are left to do that, if they're able to. And so oftentimes they're very, very obese, or end up passing away of like eating way too much, because they don't have the ability to feel full, which is terrible. Angelman syndrome, on the other hand, generally these children have like much more severe intellectual disability. A lot of times they have seizures. What else? They can have kind of like, it can kind of present similar to autism, like being nonverbal or things like that. Some people say they have inappropriate laughter, like kind of behavioral, inappropriate behaviors, things like that. So basically just showing that two completely different diseases come from the same genetics. Yeah. Okay. Last thing that we are going to talk about today is another kind of application of epigenetics, and that is X-Chromosome Inactivation. X-Chromosome Inactivation. Alright. So, over here, we've got a mama and a daddy. Females, as you know, have two X chromosomes. Maybe you don't, now you do. And males have an X and a Y. And just like any other chromosome in your body, pass on is an X. She's got to give her child an X. So we'll say she gives the child the pink X. The dad can give an X or a Y. If he gives a Y, you get a boy because you get another XY. But in this case, he is giving the blue X. And so we get a child that now has a pink and a blue X. Well, something you might not have known about females about having two X chromosomes is that in every cell of the body, one of them is active and the other is inactive. And essentially the reason for that is, think about it like, I have two X chromosomes. A boy only has one X chromosome. So there's some imbalance there. You know, I have more X chromosomes than you. And so this is the way that the body kind of handles that. Dosage compensation is what it's called. And makes it so that each cell in your body actually only has one X chromosome being expressed. And that is completely random. It's totally random which one it happens to. And so some cells will have one inactivated and others will have another inactivated. The main thing I will say is that X inactivation happens like very early in development. I think it's like right when like a female zygote is developing. And so once it happens, like it is always like that in all of your cells. So it's not like you're just sitting here and like one of your X's will randomly like switch to being active. It's like always the same in each cell. But it's random to decide like which cells. Does that make sense? I don't know. But basically what's being shown here is like this is now this baby, and some of their cells have the pink one and some have the blue one, and then the other one is still there. It's still there, but it's just a dot because it's not actually active. It's we'll talk about that, but yeah, it's not active. And so you get a girl that is part pink and part blue. Obviously not how it works, but just a visual. So the way that this happens is through epigenetics. And so here we've got like our original zygote, this girl, the zygote that made this girl, and or one of the cells that makes up her, I guess. And she's got the pink X that she got from her mom and the blue one that she got from her dad. And it is totally random that in some cells, the pink one is gonna get methylated. There are those methyl groups that we talked about before. So the pink one is going to get all of those methyl groups, and therefore it's going to be inactive. And the blue one is still going to be active. So here you can see that now all of the cells that come from that one will have the blue one active and the pink one not. And the one that's not active is called a bar body. Not important, but it really does look like that. That like there'll be an X, but then there'll be like a little dot, or not an X, but like the full X chromosome, and then just a bar body that's not being expressed. Or of course you can also inactivate the blue one and get that one methylated, and all the cells coming from that will have the pink X and a blue one inactivated. So yeah, that's X chromosome inactivation. It happens in all females. Every cell in your body only has one X active, but it's about 50%. Like some of your cells have one of them and other cells have another one. And so that doesn't cause anything specific in humans. Like we don't have any genes that make that visible, but where it does happen is in calico cats. Calico cats, it does have an effect. And so the reasoning is because the color gene, the gene that leads to the color of a cat, is on the X chromosome. It is located on the X chromosome. And so what happens in has a gene for black, but he only has one X chromosome. So he can't, he's either orange or he's black. So here he's got a Y. Ignore it, doesn't matter. But he's got an orange X, and so he's going to be orange. However, in a female, because the color gene is on the X chromosome, she could have one that has the black gene and one that has the orange gene. So then, when her chromosomes are randomly inactivated, you actually get what we're looking at over here, and you get a partially black and partially orange cat. Calico cat. And so all calico cats are female, because if you see a calico cat, it means it had some X chromosome inactivation, which means it's gotta be female, because it has two X chromosomes. So that's how you get that. Again, doesn't really impact things in humans, but yeah, you get a calico cat. I guess a couple important things to mention. First one is that all except one X chromosome in everybody is inactivated. So normally that means males don't have any inactive X's and females have one inactive X in every cell. But there are chromosome conditions where females have 3 X chromosomes, or males have 2 X's and a Y, in which case the rule is all but one X are always inactivated. So in a female with triple X syndrome, 2 would be inactivated in every cell. In a male with Klinefelter syndrome, 1 X would be inactivated in every cell. So, yeah, all but one are inactivated. The other thing I will say is the one time that it can have an effect on females, it's not with our coloring. We are not orange and black, but I've talked about it before, that X-linked traits, X-linked diseases, males are way more likely to have them because they only need to inherit one mutation on their one X chromosome in order to get the disease, because they have nothing that can overshadow it, that can make up for it. So they will have the disease if they have one mutated X, whereas for a female, if she gets one mutated X, but she has a normal one, she's fine, because that normal one is taking over. So this goes for things like color blindness, muscular dystrophy, hemophilia, male pattern baldness, stuff like that are X-linked recessive traits. And so females very rarely have them because they would have to get 2 mutated X's, like one from their mom and one from their dad, which means that their dad would have to be affected by the disease. Like, he would have to have color blindness or muscular dystrophy or whatever. However, females can show like mild symptoms of those diseases if their X chromosomes are if the inactivation is skewed in one direction. So like if the inactivation happens and 80% of their cells are expressing one chromosome, and that chromosome happens to have the muscular dystrophy gene or mutation, and then 20% are expressing the normal one, that female is probably going to have some symptoms of muscular dystrophy because the 20% of normal cells aren't enough to overpower the mutated one, if that makes sense. Yeah. I think that's all I wanted to talk about. So, yeah, I hope that you guys enjoyed this. I really had a lot of fun talking about genetics stories about what you learned and what you thought was cool and stuff, so please let me know. But thank you for watching, and I will see you next time. Let me know if you want me to do more genetics videos, or if you have any ideas. I'm sorry, genetics is like all I know. So someone commented last time and was like, can you do quantum physics next? And I was like, no, I can't. Yeah, try to keep it in the genetics or like biological sciences realm. But yeah, sleep well, everybody.